Welcome everybody to the June ERCC webinar. I'm Roger Alexander, the Scientific Outreach Coordinator for the Extracellular RNA Communication Consortium. Today our speaker is Sanjay Tyagi from the Rutgers, New Jersey School of Medicine Public Health Research Institute, who will talk to us about the molecular beacon technology he's developed to visualize RNA traffic and dynamics. Stage 2 of the ERCC's focus is technology development for doing a better job characterizing xRNA molecular carriers and isolating single EVs. And Dr. Tyagi is working with one of the ERCC2 teams led by Ionita Garan at Beth Israel Deaconess Medical Center. Dr. Tyagi has said he'll leave a lot of time for questions at the end. If you have questions during the talk, put them in chat and I'll ask those questions first. And then we'll open up the microphones. Please, when you talk, unmute, ask your question, get a response, and remute again so we avoid audio feedback. Sanjay, we're really looking forward to hearing about your work.
Right. Thank you very much for giving me the opportunity to speak in your forum. I should characterize the work I'm going to describe to you as sort of a little bit old and dated, but I think that doesn't decrease my excitement about it. I'm always really happy to talk about this work, which I think you will find useful in many different contexts of your own work. I'm going to talk about molecular beacons that we developed something like 25 years ago now. And the outline of my talk today is I will describe what are molecular beacons, how do they work, what are their common applications, and then mainly focus on imaging RNAs in live cells with molecular beacons. And the key target I have for today is to address this particular question of dispersal of mRNA molecules from the gene locus in the nucleoplasm. And I will show you how molecular beacons were used to explore that, that phenomenon. And finally, a little bit about noise in gene expression. This is more, more current work from the lab. So let's dive into it. So let's just begin with a little bit of history of hybridization. And immediately after the structure of DNA was revealed in 1953, it became clear that you could use strands of DNA as a probe to interrogate the complementary DNA. And so people began to work on it. And one of the first most important work was done by Joseph Gall in 1968, and this is Joe Gall in his teenage years. And Joe Gall is among the top American scientists of the last century, and he has produced 2 Nobel Prize winning students. And so what he did was he labeled ribosomal RNA with radioactive phosphate. He basically took these frog eggs that he used to work on, incubated them with radioactive phosphate, and so most of the RNA that these oocytes, these frog eggs synthesize, was ribosomal RNA, and that got labeled, and he used this labeled RNA to hybridize to fresh oocytes, and the idea was to see where the genes for ribosomal RNAs were in the 3 dimension of the frog egg. And so he would basically take frog eggs, denature the DNA, and fix the eggs with formaldehyde. And after denaturing, he would add this radioactively labeled ribosomal RNA, and then wash the excess of these probes and image by adding an emulsion, photographic emulsion, and you get an image like this. And each spot you see corresponds to the copy of ribosomal RNA, and you can sort of see the ribosomal genes. And you can see this cluster of ribosomal genes, and that is the nucleolus of the nucleus of these eggs. And this was just the first demonstration of hybridization. And after that, there's a long, glorious history of this technique being used in all aspects of biology. And one central idea in this kind of approach is that you immobilize your hybrids, probe bound to the target, onto some kind of solid surface. Typically, it used to be nitrocellulose membrane. In this case, it is the three-dimensional structure of the cell. And then you wash the excess of the probes. So you use large amount of probes to drive the hybridization, and then you have to remove the excess of the probes to see the signal. And what we were trying to do at the time was to see, can we actually develop a signal from the event of hybridization itself, and see that hybridization has taken place without having to remove the excess of the probes. And the idea was very simple, that we will synthesize these hairpin-shaped probes. They're about 20 to 30 nucleotides long. The loop is the probe. It is going to bind to a complementary DNA or RNA. And there is a stem ranging from 5 to 6, 7 nucleotides long. And the stem brings these two dyes in close proximity. At one end, you attach a fluorescent dye, and at the other end, you attach a quencher dye, which is not fluorescent by itself. In fact, it has no fluorescence. It's a non-fluorescent chromophore. We chose a dye called dapsil, and when the two dyes are close to each other, they will be in some kind of fret distance, and the fluorescence will be diminished. And when the loop interacts with the complementary DNA, the hybrid that forms will be long and stretched, and it will lead to separation of the fluorophore from the quencher and appearance of the fluorescence. So that's sort of basically the idea of beacons. And a simple beacon looks like this, your 15 nucleotide stem loop, 5 nucleotide stem, and these were the dyes that we attached. Dapsil is a orange chromophore without any fluorescence, and it's a dye that we picked from the protein chemistry world. And then we use a fluorophore called EDANS. It's a blue fluorescent dye. And the two were chosen carefully because for efficient FRET, fluorescence resonance energy transfer, to take place, you should have the absorption spectrum of the fluorophore should nicely overlap with the absorption spectrum of the acceptor, which in this case is dapsil, should nicely overlap with the emission spectrum of the fluorophore. And so this dye had that characteristics, and you could see the molecule had just the right features. Here is the fluorescence of the buffer, very little fluorescence. You add the beacon to it, almost no change in fluorescence. And when you add the complementary DNA, complementary to the loop, you get a bimolecular interaction that gives rise to this almost 1,000-fold increase in fluorescence. So these molecules are very well quenched, and they restore to full extent. So the signal to background will be very high. And you can, you appreciate this, that the reaction takes place spontaneously at room temperature. And so this is just a picture of this reaction on a UV box. However, and we published this paper in 1996, if we could only do this kind of probes with a blue fluorophore, this will remain a mere curiosity, and there would be not much use for it, because it turns out that even plastics have some autofluorescence in blue color. You can actually see right here, the edge of the plastics are fluorescent. And so the blue color is not very useful, and we needed to have probes that you could make with the other colors. And we said, okay, we're going to try to simply see, okay, so the DABSIL, the quencher, will not quench so well these orange and red fluorophores, but let's see how much they quench. And what we found to our surprise was, no matter what color was the dye, they were all quenched almost 99-fold, I mean, 100-fold, let me say. And so color of the dye didn't quite matter. And we explored the mechanism behind this, and I'm not going to go into it, but very interesting photochemistry takes place here. And what we found was that while the FRET energy transfer depends on the spectral overlap, what happens here is a quenching that is taking place at a close distance. The two dyes are constantly bumping into each other, and they are in contact, a physical contact. And that kind of quenching is called contact quenching, and that is not limited by spectral overlap rule. So this turned out to be the most fortunate chemistry surprise we had. And with this, you can now make probes for many different colors and use them in the same reaction, in the same test tube. And I would like to say this, that these probes were immediately useful to carry out nucleic acid amplification reactions in sealed tubes. And this is probably the first real-time PCR that was ever carried out. And what we did here was we had these 4 reactions started with, I think, 1 million molecules, 1,000 molecules, and 0 molecules. And we stopped the reactions at various cycles and just measured the fluorescence at the end. And you can see just the beginning of a hallmark of real-time PCR. And with this, we were able to do many kinds of assays for once the instruments became available to do real-time PCR. Many companies adopted this technology. We licensed the technique to 80 plus companies, and they developed tests for all kinds of pathogens, for tuberculosis, for HIV, for methicillin resistance, Staph aureus, and currently number of COVID tests, including Albert Labs, IDNO, use the same technique. And so I'll just take a moment to describe one of these assays for tuberculosis that we sort of helped develop. And basically this, and this would also describe to you the features, these multicolor aspects of the beacons. So we developed a test for tuberculosis, and we amplified the RNA polymerase, a portion of RNA polymerase gene of MTB. And when the TB becomes drug resistance, it turns out that it develops mutations in this region, which correspond to the active site of RNA polymerase. And in absence of the drug resistance, this region has almost no variation. So we had a very simple situation that we will interrogate this region with 5 beacons. And if you get a signal, you have a wild type. If you don't have a signal, you have a mutation in the region of the probe. And we could show that wild type gave signals in all colors, but these mutants, one of them will drop out. And And this way you could actually detect drug resistance in TB in less than 3 hours, while it used to take about 3 weeks to do this. And you would appreciate this, that these tests are now currently in use worldwide to diagnose drug resistance in TB. I'm going to leave this aspect of beacons of clinical diagnostics here, just give you a flavor. But what we were really excited right in the beginning was to see, can we use these probes to detect RNA in living cells? Because all you need to do is inject these probes into a cell, and they would bind to a complementary RNA, which is single-stranded. Perhaps it would be difficult to detect DNA because it's double-stranded, but RNA should be possible. And this is back in 2003. And we chose a system in which it was known that the target RNA is not distributed homogeneously in the cell, but is localized in a particular sort of portion of the cell. And so, and this thing was described by Ruth Lehman in early 90s, that fruit fly eggs have number of RNAs that are localized even before the fertilization takes place. And these RNAs will decide the fate they would decide the geometry of the organism when it develops. So for example, this RNA called Oscar is localized at the anterior tip of the growing oocyte. And this would decide that this end of the organism will be the head of the fly. And then there are other RNAs that are localized here. So we made probes, number of probes for Oscar RNA, and we would inject them into this egg. Just a little bit description of the oocyte development in fruit flies. So basically, it starts out as a single cell, and through 4 equal divisions, you have 16 cells. And one of them, for reasons we don't understand, decides to become the egg of the fly, and it will grow, and the other cells, other 15 cells, their role will become to nurse this future egg. And so this grows in size, and others will shrink. But all the RNAs and proteins and perhaps metabolites that are needed are made in these nurse cells. So they make all the RNAs, including Oscar RNA, and send it to the egg. And so we said, okay, we're going to inject beacons into that egg. But before this, we decided that if we had a beacon made from DNA backbone, it's going to bind to RNA, and the RNA-DNA hybrid that is produced will become a substrate for ribonuclease H. And that will be bad because the target RNA will be cleaved and destroyed in the process, and the beacon will return to its closed state. And so we said that to ourselves that a DNA beacon is not very good, so we modified the backbone of the beacon. So every nucleotide at the second position in the sugar had a oxymethyl group in place of a hydrogen group. And this modification does 2 things. One, the probe can bind more avidly with a higher affinity. And second, it is not a substrate for ribonuclease H. And so we made such probes. We tested them out in vitro on the synthetic RNA, and then we sort of injected one of the beacons into the oocyte, and you can see the signal right at the posterior tip for Oscar RNA. And also these proximal nurse cells became fluorescent. I said they are the ones who actually make the RNA. This was very satisfying, and we decided to say, okay, we're going to inject the beacon now into the nurse cell and ask, can we monitor the transport of the RNA to the posterior tip? And it turns out that, let me go back for a second. Oh, no, it's okay. So this movie is about 1 minute long, but it represents about 95 minutes of time in the development of the fruit fly oocyte. And so we injected in this nurse cell, and you see the beacons sort of tend to go into the nucleus, and RNAs are produced here. It binds to those RNAs, and now we know that there are clusters of Oscar RNA, and they get transported into the middle of this oocyte. And towards the end of the movie, you will see they become settled at the tip of the egg. And I know you're beginning to see it's about, I think, 60 minutes into the development. So before this experiment, there were theories about how this RNA moves. And the theory that was sort of accepted and turned out to be true is that, that there are these microtubule networks that are laid in this oocyte, starting from here, ending to the tip. And the Oscar RNA travels using a kinesin motor on these microtubules. You can see it's moving. So basically, this is the organization of the oocyte. Negative ends of microtubules, positive end of the microtubules, and these represent RNAs bound to kinesin motor. So they just dump it to the front end of the oocyte where it gets tethered. And so I'm saying all this just to say that a value of a live cell experiment confirm the idea about how microtubule network is laid, and how long does it take to do this this transport process. So we published this in PNAS, and it was really received with great enthusiasm. We had coverage in Nature and so on, but it turns out that we soon ran into difficulty. We said, okay, we're going to try to look for RNAs in cells in culture. And we found that there was weird phenomenon. First, when we injected beacons into the cells, nucleus became fluorescent. And that was very weird. And so we did carefully controlled experiments where we said, okay, we're going to take a beacon analog, which doesn't have a quantum. So just a regular oligo with a fluorescent dye with a hairpin. And we injected into the HeLa cell, and you can see how rapidly all the fluorescence went into the nucleus and stayed there. And basically, it turns out that other folks in the had discovered this, and antisense groups who were developing drugs using antisense had seen this kind of phenomenon. And basically, nucleus has in it a very high capacity to bind to negatively charged DNA. And and they are nucleopores, and they don't stop these small molecules like oligos. They go in, sink in there, they bind to histones, etc. And so nucleus acts as a sink for the beacon. So if you're going to image RNA in cytoplasm, this is not going to work. And so we decided we were going to do a trick, and the trick was to introduce a biotin group in the molecule of a beacon, and then hook it up with a streptavidin. Now it's a protein which should not be able to go into the nucleus. And when you inject that, you can sort of see for at least half an hour, they remain cytoplasm. And with this sort of modification, we were able to image beta-actin RNA in these chicken embryonic fibroblasts. And Rob Singer, earlier in the 90s, had shown that these mobile fibroblasts, they have their beta-actin RNA, which is concentrated in these pseudopods. And because it helps the cell to keep most of the actin RNA in this area so that it can help in the dynamics of the cytoskeleton that is taking place at these pseudopods. So I think once we were able to keep the beacons in the cytoplasm, we were able to see this high concentration of the beacon in live cells. And however, you notice that beta-actin is very highly expressed RNA, most highly expressed RNA in the cell. And it turns out that typical RNAs are expressed no more than 10 to 50 copies per cell. And when the RNAs are moving around in a given frame of the movie, let's say your exposure time is something like 500 milliseconds, actually the RNA moves. So what should be a fine spot for a single molecule is diffused. So what we wanted to do was to study transport of RNA from the gene locus to the nuclear pores. And I pointed out to you this problem of beacons going into the nucleus and staying there. And so we decided we are going to work with that property of oligos that we are just going to image in the nucleus using simple beacons. So leaving behind this beacon tethered to a protein, we went back to beacons that were pure, no protein attached to them, and we would detect an RNA that cell would make. And what we decided to do was to engineer an RNA, a completely unnatural RNA. And the idea was that in a non-coding region of this RNA, we will have a tandem repeats of a 50-mer sequence. In this case, we repeated it like 96 times. So there's this long array of these sequences at the untranslated, 3' untranslated region of a GFP coding sequence. And we created this artificial gene and we stuck it into the nucleus of Chinese hamster cells. And why are we doing this? What purpose will it serve? So, let me transition to this slide here. If you consider the human nucleus, a nucleus of human cells, it has about 5-meter long DNA. And this long DNA is stuffed into a volume with a diameter of about 5 micrometer. So very dense matrix in that space. And DNA is, as you know, is not naked. It is complex with histones and other proteins. So it creates a very, very dense, physically dense material. And scientists, cell biologists were concerned, how does RNA that is created at the gene locus travel through this dense matrix? And RNA, when it is made, during the transcription binds to something like a couple of dozen proteins. So these 2 dozen proteins are bound to the nascent RNA, and this complex of RNA and protein will have to move through this dense matrix. And so cell biologists were proposing all kinds of ideas. Gunther Blobel proposed this idea that a gene, when it gets activated, actually moves to the periphery of the nucleus where it will be shuttled, where RNA can shuttle out. And Robesinger and others, they noticed that if you look at nuclei of highly productive cells, they showed some kind of tracks. So there were these ideas that there is active transport of RNA going on in the nucleus. And I'm going to come back to it. There was clear physical evidence of some kind of active transport taking place in the nucleus. So we said we have an opportunity to know, look at RNA movement in the nucleus and ask, is it active or not? So that's why we engineered this gene with tandem repeats. And so this gene not only codes for GFP, we can control the expression of this gene, and we isolated cell lines that had a copy of this gene integrated in some place in the chromatin. And you can see by DNA fish, every cell has that gene. And when you remove doxycycline from the culture medium, they produce GFP, meaning they were able to transcribe the RNA, they were able to translate that. So the gene was functional. In absence, with doxycycline, you could keep the gene off. And that allows you to do controls. So we took these cells, in fact, this very 5 cells, and we injected beacons into them. And so these beacons, like I said, would bind to, one beacon will bind to each repeat, 96 beacons binding to one RNA molecule. And you can sort of see these spot-like signals in the nucleus. And in absence of RNA expression, which happens in doxycycline, you don't see those spots. And this is a sort of enlarged area of one of the nucleus. And these spots we were able to show are actually single molecules of this RNA and bound to perhaps 96 beacon molecules in live cells. And they're moving around. They and this is only one movie I'm going to show you. This is a HeLa cells where Chinese hamster cells where this reporter is being expressed, and you can see molecules of this RNA in the nucleus as well as outside in cytoplasm. And for the rest of the seminar, I'm just going to analyze how they move and what does it tell us. So my student Arjun Raj, who was here, a math student, he was able to write computer algorithms to identify these spots and track them over time. And he was able to, you can sort of see the same cell tracks behind each molecule that was moving in the nucleus. We tracked the cytoplasm as well, but let's focus on the nucleus. And I think the gene locus is somewhere here, and these molecules are moving around. And we also expressed histone 2B as a GFP construct. And the idea was that we would be able to label high-density chromatin and distinguish it from relatively low-density chromatin. And this map, this orange color, shows expression of histone 2B. And so now these tracks can be seen in relation to the chromatin or high-density chromatin. There's chromatin everywhere, but there are areas of low-density chromatin. And what we found immediately, if you look at these movies, that most of the motion is restricted to low-density chromatin area, where GFP of histones is lower. And we analyzed the tracks a little more closely. And so here is a typical track. So basically, you can see the position of a molecule on first second, 1 second later it goes here, 2 seconds later it goes here, and so on. So you can sort of plot the location of this molecule in relation to its original place. And you can develop these sort of tables where for the same molecule, you can say, okay, but it moved about 0.13 micrometer in first second, so much in the second, and so on, and average 1 second displacement was this. You can also ask, how much does it move in 2 seconds? And it would have moved from here to here in 2 seconds, or from here to here in 2 seconds, and so on. So you can develop a table of displacement in 2 seconds, 3 seconds, and so on. And you can analyze this data, and basically the law of diffusion worked out by this guy whom I returned to, Robert, what is his name? Robert Brown. I come back to a little story about him. But basically, what you find is that time interval is proportional to the mean square displacement if the diffusion is free. And we found that most of the molecules were moving by free diffusion. If it was an active transport, it would sort of look like this. It would be like a sigmoidal curve going up like that. So it will move very fast in short period of time. And there's no evidence of active transport in most of the molecules. However, some molecules showed the evidence of confined diffusion. You can sort of analyze this graph right here that it seems to be freely moving in that space, but then beyond the space, it's confined. So those little pockets are confined diffusion. And we also found about almost half of the molecules, or let's say 25% of the molecules were stationary. They were not moving at all. And I come back to this. Let's go to Robert Brown. So it turns out that this Brownian motion is named after Mr. Robert Brown, who in early 19th century had been looking at motion of pollen grains in water. So, and he found that they continuously move. There's like mad motion with no rhyme or reason. And he described this phenomena in a couple of papers, and it became so people, the bourgeoisie in Europe was fascinated by this phenomena. They were, you would get a microscope in gift, and you would throw some pollen grains on a dish and look at them. And people were wondering, why do they move? And they said they're animated, they're ghosts, they're magnetic fields moving them around. And this reason for this motion was not explained for another 100 years. And the person to explain why these pollen grains or anything moves in this kind of motion, the person to explain the physics behind it was Albert Einstein. And basically, the reason is these little particles like pollen grains get hit by continuously moving water molecules. And that imparting of the energy from water molecules to these larger things cause them to move in various directions. And that's sort of Brownian motion. And that's exactly what is taking place with these RNA molecules. I should say one more thing about this. The diffusion constant of these molecules was they were slower than typical proteins. They're about 1/10 as much mobile. And that is rational because they're about a dozen or 2 dozen protein molecules bound to an average RNA. I should say one quick thing about here. So we found about 25% of the molecules were stuck. These red guys were stuck. But they will move again. And we found that if you deplete the cells of ATP, then more will get stuck. And if you rescue the ATP restriction, they will move again. So it turns out that people had seen this kind of ATP dependence in motion in the nucleus before, and that was the basis of active transport model. And we see the same kind of thing, that ATP depletion causes more molecules to come to stop. And we believe, and a number of other papers have been published, they say the same thing, that chromatin itself is alive. It depends, and its motion depends on ATP. So it's not the molecules of RNA or RNP that needs ATP to move, but it's the chromatin. And with that, I'm going to leave this issue of movement here and just spend 5-8 minutes on one last thing that we learned from this study. You notice that I'm going to go quickly back to this image. We injected beacons into these 4 cells that look identical. They have one copy of the gene, but this cell has, a couple of hundred spots. That has few spots. This had more than 1,000 spots. We found that as we looked at these cells more and more, they were highly heterogeneous, while they were genetically identical. We isolated them from a single cell. And when you remove doxycycline, they should all be expressing in the same level. And so my student, Arjun, the mathematician, he said, I'm going to analyze them quantitatively and not use beacons. He said, beacons are too cumbersome. I have to inject them into cells, and that's not going to work. I need to visualize hundreds of cells. And so he said, I'm going to go back to what Joe Gall used to do. Fix the cells, formaldehyde them, remove the membrane, add probes, and look at RNA. And this is what you see. And you can count in a fixed cell now all the copies of the molecule of RNA. And you see these fields where some cells have a lot of RNA, and those that have a lot typically have a cluster of RNA, which is in the nucleus, and many cells have no RNA. He developed a histogram from this data, and this is published in this really popular and hot paper in PLOS Biology 2006. And what these histograms tell you is something very interesting, that almost in this population, 55 cells make 0 to 10 copies of RNA, 9 cells make 10 to 20 and so on. Only one cell makes about 600-700 copies of RNA. This distribution is very odd. This is not what you expect from the cell. This is sort of like wealth distribution in the society. These are the Bill Gates of cellular society, and this is all of us together. So this is highly skewed towards known performers. And if every cell was likely to make RNA with equal probability, it would follow a Poisson distribution. Meaning, if the average of this population is about 90 molecules, the distribution should be something like this. And you can sort of see that and there is this statistical parameter called Fano factor, which is essentially standard deviation squared divided by mean. So this would be the width of the hair would be the standard deviation squared divided by mean. And for Poisson, it's 1. And for this population, it was 200. It departs a lot from expected Poissonian distribution. So people said, these are weird cells. These are heavily engineered. They have doxycycline controls. You have these arrays, repeats, and so on. God knows what's going on. Maybe the cells are in wrong stage of cell cycle. And so he did a very interesting experiment to address these. And he said, I'm going to create 2 such genes in the same cell. One I will be able to probe with red probes, other with the green probes. But he did the experiment in such a way that in one case they went next to each other, and in another case on 2 separate locations in the DNA. And they showed completely divergent behavior. So this is different loci, and you can sort of see the cells that were making green RNA were not making red. Those that made red didn't make green. So I want to stress that these genes are controlled by they have the same promoter, the same thing turns them on, and yet they're expressing only one of the 2 genes. And it's true statistically when you look at many cells, they either make red or green RNA. Rarely they make both. But when the 2 genes were next to each other, the RNAs were produced in a coherent manner. The cells, if it made green, it also made red. And statistically, that is true. So basically, he developed a mathematical model. And the model was that mRNA is not made in a continuous and steady stream. It's actually being produced in some kind of bursts. So you make a burst of RNA, hundreds of copies within a few minutes, and then the gene comes to a stop. It doesn't make more RNA for a while. And during that period, RNA decays, and then a new burst takes place, and then it will decay. So in a given cell, the population of RNA sort of changes over time like this, and it just follows the activity at the promoter. And the promoter exists in two states, either on or off. And this is, I would like to stress that you have removed doxycycline to turn it on. So everything should be on continuously, and yet it is not. And so this is 2006. No cell biologists have shown that the same phenomena takes place in not just cell culture, but in your body and all kind of organisms, C. elegans and so on. And one more thing I would like to point out, the variability is there in RNA, of course. So you can see a field of cells where RNA is imaged, and in the same field, we can image how much protein, GFP in this case, was made. And you can sort of see GFP, or the protein, is also highly variable. These cells make a lot of protein, these not so much. But what happens is that RNAs are being made in a burst, but they're short-lived. After making a few hundred RNAs, like this cell had stopped making RNA and RNA has decayed, but the protein product it made hangs out for a longer time. Because of the long half-life of the protein variation is buffered. It's sort of a little bit lower, while RNA variation is high. So the stochastic expression of RNA and also protein has become a major descriptor of single cells. And the reason is that, and we have a paper just published a few years ago, and I'm going to give you the model, and we can discuss if you are interested. And basically the idea is that higher eukaryotes have a lot of transcription factors, maybe 10,000 copies of any given transcription factor that cell has at a given moment, but gene is hidden from it. The genes are, the promoters of the genes are tightly sequestered in chromatin, and some kind of random breathing in the chromatin allows initial access to the transcription factor, and once it gets in, it makes a sustained output of RNA, and then because there are these other condensing enzymes that return the chromatin back to the native state, the activity stops. So gene continuously experiences these sort of on and off periods. And okay, I'm going to stop here and quickly, within 10 seconds, show you the faces of people who have done the work. This live cell experiments in the fruit flies were done by Diana Bratu when she was a graduate student. She's a professor in Hunter College and became a fruit fly biologist. Arjun Raj was the mathematician turned biologist. He's a star at UPenn. His Twitter has 10,000 people following him. Diana Vargas still in the lab. She developed these arrays of tandem repeats. It took 2 years to make one construct, Herculean task. And this is Salvatore Maras. He walks into my office with, he's an amazing chemist he is holding these beacons in his hand. And Latasa Fraser, she did the work that I just alluded to at the end. And today she's getting married in I think in half an hour. And this is Fred Kramer. He's my guru, he's my mentor, and I share a lab with him for the last 35 years. And he's also my umbrella shelter and so on. And the work was done here. And I think I have a few minutes, 10 minutes, 15 minutes, I can answer questions.
Sanjay, thank you so much. We will definitely get you out of here on the hour or before if you need to support your lab member. That's beautiful, beautiful work. One question from Mina they ask, does binding of the beacon interfere with proteins already bound to RNA in live cells?
Yeah, it could. So, I mean, you probably have seen pictures of RNA, electron microscopic pictures of RNA in cells that are just busted open, and they look like necklace, bead necklace, meaning entire RNA is coated with proteins. So those proteins are bound. However, RNA binding proteins are typically bound weakly. And these probes, as you would imagine, are going to make concerted binding, 20 nucleotides. And they're going to displace those proteins. And it may have some adverse effects or may not. So in our case, we targeted UTR, untranslated regions. So they should not be adversely affecting. But they do, I think in live cells, they can displace the proteins for sure.
That leads me to my very basic question. On Dana Vargas's 96 MERS, can you, I don't fully understand how the beacons bind to the repeats. Can you go over that again?
Yeah, sure. So basically, one beacon bind to one repeats, and therefore one RNA will attract 96 beacons. So like here, and these are 50 mer repeats. So we took special care that the adjacent beacons don't quench each other. If they were like, next to each other, it could be that this fluorophore will be quenched by that quencher. So we made them deliberately long. So that's how. So basically, large number of beacons binding to the same molecule.
And can you go back to the image showing the beacon itself? Is it the beacon itself forms a stem loop and creates fret between its two ends? Is that what's happening?
Yes. So basically, the stem brings the two dyes next to each other. So I'm showing you here. This is not to the scale, right? The two dyes are really close. They are within 20 angstrom of each other.
So that entire thing is the beacon.
That's right.
And where would, is it in the loop that would bind to the repeat of the target mRNA?
That's right.
Okay.
So, however, I think in those cases, we chose that one of the stem would also bind to that. So you see this, we did a trick there that, so this picture shows that this is fully bound there, right? So one of the stem was also binding to the repeat. So repeat and the beacon sequence was chosen purposely because of this issue of neighbors quenching each other, right? We want to avoid that. So this way they would not have neighbor to neighbor quenching.
Okay. And let's see. Where's the other question? Oh yeah, M. Kumari says, this is very interesting and can help explain. This was on the mRNA motion portion of the talk. And they said, this is very interesting and can help explain transfection efficiency. Maybe you want to comment and maybe they want to expand on the comment.
Hi, this is Nina here. It's a very nice talk, Dr. Tyagi. I really enjoyed. My comment was to, where there is a differential expression of RNA, that RNA is either, it is transcribed intermittently, not continuously. So when we do the transfection, we always talk about the efficiency. So, it's always very low. So if the transcription is intermittent, that indicates that why we have low transfection efficiency. It may not be the efficiency of transfection, but it's actually the expression of the transgene in the cells.
Yeah, this is very, very interesting thought. So we calculated the sort of interval between these bursts, right? And for natural genes, which are nicely chromatinized, gene is hidden in the chromatin, the bursts happen about, like 15-minute time scale. the interval between the bursts is about that much. So if you are waiting for one night after transfection, it should be able to produce your transgene product in that that period. And also additional thing about the transfection is that your plasmids are not chromatinized, meaning they're sort of naked DNA, and you put a lot of them. You know, it turns out that in transfection you probably put 100 to 1,000 copies of the plasmid in the same cell, and it's heterogeneous because some cells get only a few, some get a lot, and that's sort of nature of transfection. And on top of all this could be this heterogeneity from the transcription itself. But I think there are multiple steps here.
Thank you.
The same person asks, are plasma membrane diffusible beacons available?
So I think Ionita has done experiments with these cell penetrating peptides that are hooked to beacons, and together they were able to take the beacon into the cells. We actually, our work is sort of early, and we tried to do other means of introducing beacons, but we found that if you're going to do imaging afterward, things like transfecting beacons, things like cell penetrating peptides gave us sort of confusing images. And that this injection, which is tedious, but it gave us sort of very clear images that we could interpret.
I would note that Ionita Garan is going to give an ERCC webinar later this year, perhaps September, to show how his lab has applied these technologies to work.
I hope I can join in too, right?
Yeah, absolutely.
Okay.
Ahmed asks, have you tried different lengths of stem and or loop?
Yeah, many different many different stems and loops. So loops are they range from like something like 12 nucleotide to 30 nucleotide there, that depending on the situation you can change it a lot. And the stems are typically 4 is low end, 5 to 6, 7 nucleotides. If you make them very strong, it will be difficult for them to open spontaneously, because it's an intramolecular hybrid. Even though it is it's only a short hybrid, it is difficult to break when it's intramolecular. So it should not be longer than 6, 7 nucleotides. And of course, in PCR, high temperatures experience, there you could have higher, stronger stems. I have, and I can send these very simple design, rules to folks if they're interested in design. And I help people design beacons all the time.
Great. I'm happy to mediate that if anybody wants to. info@xrna.org, and we can. We may be doing a workshop on molecular beacons at some point, so stay tuned for that as well. Let me ask one final question, and you can say, I gotta go and not answer it because I know you have somewhere to be. In the fruit fly oocyte movies where you showed the RNA in the nucleus and then it transferring to other cells, it seemed as if the fluorescent signal disappeared when it was not, like it reappeared in the nucleus of the other cells and you didn't see it in between. Do you understand why that happens?
No, I think it could be simply that over this long term, we sort of were in different part of, we were not focusing on the center. And so it is true that there is some kind of low light time period, and then it becomes bright again. I don't think there is I think it's just the artifact of the imaging.
We should leave it there, and congratulations to your lab member, and thanks very much for your time today.
Thank you very much. I really appreciate it. Thank you.